Hello and welcome to my presentation on cellular respiration and how it can generate 30 ATP and therefore be a very effective process in the body. So I'd like to start off talking about glycolysis, and so we'll talk a little bit about the different stages where ATP is involved, whether it be directly in the form of ATP or if it's through a conversion ratio with NADH. And then we'll talk a little bit about what the conversion ratios are and kind of give an example here using the different steps of glycolysis. Then we'll talk about two other forms, citric acid cycle and the oxidative decarboxylation of pyruvate. And then we'll kind of go through just step by step as to how we get those 30 ATP. So first, the step that we'll talk about here in glycolysis is glucose to glucose 6-phosphate, and we can see that ATP becomes ADP, and so we actually consume an ATP when we're in this step, and that's because hexokinase is involved, and kinases are enzymes that catalyze the transfer of a phosphoryl group from ATP to an acceptor. So it takes the triphosphate and makes it a diphosphate, because it transfers it to an acceptor. Then the next time we see it happening is actually here in step 3, where we see fructose 6-phosphate go to fructose 1,6-phosphate, and that's done through phosphofructokinase. Again, it being a kinase will therefore catalyze that transfer of the phosphoryl group of ATP. So again, we see a consumption of ATP. And then when we jump down to step 6, we see glyceraldehyde 3-phosphate being converted to 1,3-bisphosphoglycerate, and that involves NAD plus to NADH via glyceraldehyde 3-phosphate dehydrogenase. And as we know, dehydrogenases are enzymes that catalyze oxidation reduction reactions and often transfer a hydride ion from a donor molecule, such as NAD+, or transfer a hydride ion from NADH to a acceptor molecule. In this case, it's NAD+ to NADH. And we'll talk a little bit about the conversion ratio, but there is ATP involved in that as well. And then we see from step 7, 1,3-bisphosphoglycerate to 3-phosphoglycerate produces actually 2 ATP from 2 ADP, and that's catalyzed through phosphoglycerate kinase, which is done using what's called surface level phosphorylation. That's the word I was looking for. And essentially what that means is there's the kinase substrate has a high phosphorylation potential, transfer potential, and so it will transfer. And then we see it again happening in step 10, where we see phosphophenol pyruvate convert to pyruvate. And again, we see the conversion of 2 ADP to 2 ATP. So when we talk about kind of calculating ATP, we want to look at the conversion rates here on the left. So whenever we see NADH being NAD plus being converted to NADH, will yield approximately 2.5 ATP. When FAD is converted to FADH2, that yields about 1.5, and ATP, of course, is itself, it yields 2. So looking at glycolysis, where we just looked, we can see the different steps, whether they convert, how that converts. So step 1, ATP is lost, so it's a -1. Step 3, -1. Step 5, we see ATP being converted, so that's 2. Step 6 is 2, and then step 10, is 3. So that's for a total of 5, because we lose 2, so we gain 3 plus 2 is 5. And then in order to enter the citric acid cycle, pyruvate has to convert itself to acetyl CoA, which is kind of a long process, as we've learned already, but we can see here that NAD plus converts to NADH, and as we mentioned already, when that happens, we yield about 2.5 ATP, so that would come for a total of 5. Then it can enter the citric acid cycle here, and so we'll consider this citrate synthase kind of step 1, and then aconitase would be step 2, and then so on and so forth until we get to isocitrodehydrogenase, where we see NADH plus through dehydrogenase being converted to NADH. And then we'll continue on to where we see alpha ketoglutarate convert to succinyl CoA, where again we see NADH plus becoming NADH. Continuing on, succinate to fumarate, FAD to FADH2, and then finally malate to oxaloacetate, we see NADH to NADH plus. So we'll talk a little bit about the conversion. So overall, how do we get the 30? Well, we already talked about glycolysis, right? We saw that it produced a total of 5 ATP, because we have the -2, and then the other ones, this should be step 10, sorry. And then the oxidative decarboxylation of pyruvate, we saw that it was involving NADH, so we have the 2.5 conversion here to 5, and then in the citric acid cycle, we can kind of run through it here, but FADH, as we know, produces that 1.5, so this is where FADH is. These are NADH, or FADH2, excuse me, and these are NADHs. And then this is ATP production for a total of 20. So we have 5, 5, and 20, thereby being 30. Thank you for listening.